Hello, this video is going to focus on cell division and a few other things that cells do in general. And towards the end of the video, we will put what we learned in the context of stem cells. First, let's define some terms. Cell division refers to the process of one cell dividing into two. For a cell to divide, it has to grow larger and make two copies of everything, including DNA, which will be in its condensed chromatin form. Once everything is prepared, the cell will begin to physically divide in a process called mitosis. Cell division and mitosis are sometimes used interchangeably, but cell division is more general and includes the entire process of growth and preparation, while mitosis is only the part where the cell actually divides into two cells with identical copies of DNA. Just as a brief overview of mitosis, here is a cell that is ready to divide and has two copies of its DNA. Mitosis begins when these two copies pair up during prophase. I only drew one here, but really there should be 23 of these. The pairs then line up in the center of the cell during metaphase. This is also when things called mitotic spindles form, and those are made out of microtubules. The microtubules then pull the paired chromatids away from each other during anaphase, and the cell begins to pinch into two during telophase before they finally completely split up into two cells. Cell proliferation refers to an increase in the number of cells, and although no one really calls it this, it's essentially cell multiplication. Since cells can only increase their numbers by dividing, it can be pretty easy to mix up cell proliferation and cell division. Ultimately, the result is the same, since if you divide a cell, you end up with more cells. So it's just that division specifically refers to the preparation and act of splitting, while proliferation is about the number of cells. Either way, cell proliferation requires cell division. Cells are probably the only context in which multiplication means the same thing as division. Apoptosis refers to cell death, and is usually specifically referred to as programmed cell death. This is because one of the reasons why apoptosis occurs is that the cells are no longer needed, so they have been instructed to die. One example is during development, when cells that make up webs between fingers die so that the fingers are no longer webbed. Apoptosis can also occur when cells get too old and need to be cleared out, or when the cell realizes there's something wrong and decides to die before it divides and creates more cells with the same problem. This is very important because if a cell obtains a bad mutation and it isn't able to undergo apoptosis, this cell will create more cells with that same mutation, and over time, this collection of cells can get more and more mutations and eventually become cancerous. We'll learn more about that later. Cell differentiation refers to a cell becoming more specialized. In other words, the cell is taking on characteristics that differentiate it from other types of cells. For example, while keratinocytes and cardiomyocytes look and function completely differently, they all have the same set of DNA. Differentiation is very much a spectrum, with cells that haven't chosen what they wanted to be yet being the least differentiated, cells that have become what they want to be being the most differentiated, and cells that have committed to a path but haven't quite finished falling somewhere in the middle. Now let's take everything we've learned and see how they apply to stem cells. I think that stem cells are super cool, and hopefully you'll think so too. Stem cells are cells that have the potential to become other types of cells. You can also think of them as a cell that other cells stem from or are derived from. Their potential to become multiple types of cells is referred to as potency, and there are a few different types of potency. Embryonic stem cells have the potential to become any kind of cell in the body, and this makes it a pluripotent stem cell. These stem cells are able to form keratinocytes, cardiomyocytes, neurons, etc. These are the least differentiated types of cells. You and I also have stem cells, but the only stem cells we have inside our body are called adult stem cells. While they can become many different types of cells, they can't become anything. This makes them multipotent stem cells. They aren't the least differentiated, but they do fall close to that end of the scale. For example, we have blood stem cells, also known as hematopoietic stem cells, with hema like heme, meaning blood, and poietic meaning producing. These cells can't become keratinocytes, cardiomyocytes, or neurons, but they can become red blood cells, B cells, and all the other cells that belong to the immune system. Another special characteristic of stem cells is that they can make more of themselves indefinitely, with no limit on the number of times they can go through cell division. They can also do asymmetric cell division, which means that when they divide, one cell will still be a stem cell, while the other cell will be more differentiated. So if we look at a non-stem cell, when it divides, it will make two cells that are exactly the same as each other and as the original cell. Furthermore, these cells can only divide so many more times before they eventually lose the ability to continue dividing. On the other hand, stem cells can make more stem cells while also making other cells that are more differentiated. The new stem cells can continue to divide indefinitely, as represented by this arrow, while the differentiated cell cannot. Progenitor cells are very similar to adult stem cells, but there are a few differences. Like adult stem cells, they can become multiple types of cells. However, these are cells that have begun to commit to a specific path and type of cell, so they are even more limited in the types of cells they can become, making them fall more to the right of the adult stem cells on the differentiation scale. Another difference is that they can only divide a limited number of times, like other non-stem cells do. Going back to the example of hematopoietic stem cells, there are multiple differentiation steps required before it becomes a red blood cell. The cells at these steps in between can potentially become other types of immune cells, but with each differentiation, they become more and more committed, until finally they become a red blood cell. While the hematopoietic stem cell still has the potential of going down any of these paths and can, for example, become a B cell down the line, the cells that have committed to the path towards becoming a red blood cell still has other choices it can make, but its path to becoming a B cell is now lost. Note that stem cells generally go through a progenitor cell state. They don't immediately become fully differentiated after just one round. So now you know what makes stem cells so special, and also know a bit more about the life of a cell. That's it for this video series on cells, and thank you for watching.